Hi everyone. Last time we have already introduced the free energy difference to accelerate the rate of a reaction. However, so how can we explain the rate enhancement in terms of thermodynamics? So today's topic is to introduce the transition stage in enzyme catalysis. So you will see here there is a transition stage existence during the reactions showing in the figure. So the transition stage is a transitionary molecular structure that is no longer the substrate, but is not yet the product. The transition state is the least stable and the most seldom occupied species along the reaction pathway because it is the one with the highest free energy. The difference in free energy between the transition state and the substrate is called the Gibbs free energy of activation, or simply the activating energy, marked as delta G. Thus, we see the key to know how enzymes operate. And enzymes accelerate reactions by decreasing delta G, the free energy of activation. And the essence of catalysis is facilitating the information of the transition state. So how to form the transition state? Much of the catalytic power of enzymes comes from their binding to and then altering the structure of the substrate to promote the formation of the transition state. Thus, the first step in catalysis is the formation of an enzyme substrate complex, short for ES. So it shows here that an enzyme substrate complex, ES complex, left is enzyme cytochrome P450, and it is bound to its substrate chamber as shown in the right. What is the evidence for the existence of an enzyme substrate complex? Well, firstly, the first clue, also the most general evidence, was the observations that at a constant concentration of enzyme, the reaction rate increases with increasing substrate concentrations until a maximum velocity is reached. And we could find that at a sufficiently high substrate concentration, all the catalytic sites are filled, or we call it saturated, and so the reaction rate cannot increase. The fact that an enzyme catalyzed reaction has a maximum velocity suggests the formation of a decreased ES complex. Furthermore, the spectroscopic characteristic of many enzymes and substrate changes are also shown the formation of an ES complex. So it shows here that fluorescence remarkably changed when tryptophan synthesis catalyzed the synthesis of L-tryptophan from L-serine and an indole derivative. So this figure shows that X-ray crystallography has provided high-resolution images of substrates and substrate analogs bound to the active sites of enzyme. As you can see, there is a region in the enzyme that could combine with the substrates we called active site. And there is several common figures of enzymes' active sites. Firstly, the active site is a three-dimensional cleft, or is formed by groups that come from different parts of the amino acid sequence. And it shows up here that the ribbon diagram of an enzyme lysosome with several components of the active site, and it's shown in color. So also in this lysosome is shown in the right schematic representation, and the important groups in the active site are contributed by residues numbered 35, 52, 62, 63, and 101, and 108 in the sequence of 129 amino acids. So Second, the active site takes up a small part of the total volume of an enzyme. And the active sites are unique microenvironments, a nonpolar microenvironment. At the same time, substrates are bound to enzymes by multi-weak attractions, such as hydrogen bonds, from the figures shows here, and it found that enzyme ribonuclease forms hydrogen bonds with the uridine component of the substrate. Finally, because of the enzyme and the substrate interact by means of short-range forces that require close constant, a substrate must have a matching shape to fit into the site. There are two typical models of enzyme-substrate interaction. One is the lock and key model of enzyme-substrate binding. And in this model, the active site or the unbounded enzyme is complementary in shape to the substrate. So the other one is induced fit model of enzyme-substrate binding. In this model, the enzyme changes shapes on substrate binding, and the active site forms a shape complementary to the substrate only after the substrate has been bound. Therefore, the common figures of active sites are above mentioned. So next, as we know, enzymes lower the activation energy. But where does the energy to lower the activation energy come from? The free energy released on binding is called the binding energy. Also, the transition state is too unstable to exist for long, and it is determined by the delta G of the reaction. However, the maximum binding energy is released when the enzyme facilitates the formation of the transition state, lowering the activation energy. So, the binding energy between enzyme and substrate is important for catalysis. Okay, so that's all for today, and thank you for listening.